Welcome everybody to this plenary colloquium of the Italian Society in Statistical Physics. So it's a great pleasure for me to introduce Professor Nicolas Brunel. He's professor at Duke's University in neuroscience and neurobiology and physics. He graduated at the NS in Paris. He has been spending a couple of years when we were a little bit younger in Rome as a postdoc, has been a researcher at the French CNRS, and then he has been professor at the University of Chicago before going to Duke's University. And his talk is about models of memory, its insights from statistical physics. Thank you.
Well, thank you very much. So first, I'd like to thank Luca for the invitation to give us a seminar here. I'd like to thank also Anzo and Giorgio for their hospitality during this year, where I'm kind of traveling around between Italy and North Carolina. And for me, it's always a great pleasure to be here in Rome. I have great memories of the time when I was here in the 90s working as a postdoc with Danny Amit. And for me, these years had really a very deep impact, both on my personal life, but also on my professional life. And it's always a great pleasure to come back here. So I'd like to tell you about models of memory in networks of neurons and what can we learn from using methods from statistical physics to understand the properties of these networks. And I'd also like to make connections with data in neuroscience. So you'll see both methods from statistical physics, but also neuroscience data, and I'll try to explain some of the details of this data. So let me start by a model that had a tremendous impact on the field, which is the model that was proposed by John Hopfield in 1982. So this is a model that's probably familiar to many of you, but just in case, I thought I could start by explaining this model. And so this is a model of a large network of n neurons, and Hopfield chose to use a very simple network where neurons are spin-like variables. So plus or minus one. Plus one means neuron is active. Minus one means that it's inactive. And then neurons interact together through a synaptic connectivity matrix Jij, which would be the analog of the couplings between spins in a magnetic system. And so this would be the total synaptic inputs that a neuron receives. And if these total inputs are above the threshold, which here is just 0, then the neuron will be active at the next time step. So at this point, this is basically, there is nothing different from a kind of a standard model like the Ising model, or but the specificity of this model is how the synaptic matrix here is structured. And so Hopfield was wondering, how can you store information in this network? And so he considered information in the form of a random IID pattern. So the memories that the network has to store are these random patterns where psi I mu is the state of a particular neuron I in a particular memory mu. And here, this is a cartoon of this model where we have just 4 neurons with different colors. This indicates the connectivity matrix. This would be the axon of a neuron that connects to the synapses of all other neurons in the network. Note that there are no self-coupling, no autopsies. And here is a particular pattern. The idea is that these patterns are imposed from external inputs. So so here is a particular pattern in which these 2 neurons are active and the other 2 are inactive. So how to store this in the connectivity matrix? Well, Hopfield used all the ideas that were introduced by a Canadian psychologist, Donald Hebb. And Hebb hypothesized that if 2 neurons are active together, then the synapses between these 2 neurons would tend to strengthen, to become stronger. And so Hopfield thought that, well, a simple way to store information would be to modify the connectivity matrix by changing the weights in proportion to the product of the states of the pre-synaptic neuron psi j mu and the post-synaptic neuron psi I mu. And then you can just use a linear superposition of these synaptic modifications for all these patterns, and that gives you this connectivity matrix. So here we see that, for instance, we have this particular pattern that is presented, and to store it in the connectivity matrix, you just strengthening the weights between these active neurons, and vice versa, you're decreasing the weights connecting these active neurons with all other neurons in the network. And then you can go on, show another pattern, and then and continue structuring the connectivity matrix in this way. And of course, since the connectivity matrix is symmetric, there are no self-coupling, so you can define an energy or a Lyapunov function, and then you can use the standard toolbox from statistical mechanics. And that was done a few years later by Amit, Gutfreund, and Sopolinsky that derived this beautiful phase diagram where on the x-axis you have the number of patterns that you're storing the connectivity matrix P divided by the network size N. So this is this alpha is the ratio between P and N. And here, let's focus on this line here, Tm. So this is the temperature. What I've been describing so far is just the dynamics at 0 temperature, but you can also introduce a temperature. And in this whole region below the line Tm, what they showed is that you have the presence of so-called retrieval states. So there are local minima of the free energy that are strongly correlated with all the stored patterns. And so the space of states of the network, you can think about it in this way. You have these minima of the free energy that are these points here, which are strongly correlated with the stored patterns. And these are typically large basins of attraction, especially if the number of stored memories is sufficiently far from these critical lines. And what they computed is the storage capacity, so the maximal number of memories that you can store in the system, and they show that basically you could retrieve, store and retrieve memories in this network if the number of stored memories is less than 0.138 times n. So you have a capacity that's extensive in the system size. So this model has a tremendous impact on the field, but of course this model is very far from real neurobiological networks. And so in the following years, many investigators studied various generalizations of this model, and in fact, they show that the basic properties of this model are basically robust to many of these modifications. And here I just there is a very long list of generalizations. This is only a partial list, of course. I just want to mention two of them. So in particular, Derrida, Bernard Derrida and others with Zipelius and Gardner, they show that if you dilute this connectivity matrix, then what they showed is that you can still store and retrieve memories. And interestingly, the storage is actually more efficient in a sparse network. If you go to the sparse limit, the capacity, the number of patterns that you can store per available synapse is actually larger than in the Hopfield model, which is a quite interesting result. And the motivation here is that in real neurobiological networks, neurons are not connected all to all. There is typically a connection probability, which is typically in cortex or hippocampus structures that are thought to be involved in memory. They are about a few percent or maybe 10% or something like that. Another generalization that goes towards more neurobiological realism is the one introduced by Mishat, Sodix, and Feigelman. And the idea here is that instead of the Hopfield model, where in the Hopfield model, a neuron is active in average in half of the memories, and that's very unrealistic. Typically, a neuron will be active only in a small fraction of inputs that you can present to the network. So it's thought that encoding is rather sparse. So this model generalized the Hopfield model to sparsely encoded memories. And again, here they show that the capacity actually increases as the coding level F decreases, and it actually diverges in the sparse coding limit. This parameter alpha, the maximum capacity actually increases as 1 over 2f log of 1 over f. And there's been many other generalizations. I just want to mention here models, particular one influential generalization by Giorgio, who showed that a particular way in which you could learn new patterns continuously while at the price of forgetting old patterns. So this is in fact one example of a class of models called palimpsest models that were also studied by other authors. And I also want to mention that when I was here working with Dani Amit, we also worked on generalizations that with models with much more biophysically realistic architectures and neurons. And to conclude this review of this literature, I'd like also to mention a completely different approach that was introduced by Elizabeth Gardner, the late Elizabeth Gardner, in the late 80s. And so all the models that I've described so far study the dynamics of networks that have a fixed specified connectivity matrix. What Gardner thought is that rather than focusing on a particular plasticity rule, it would be nice to study the space of all possible connectivity matrices that satisfy constraints imposed by learning. So the idea is that I give you a set of patterns that you have to store. So this imposes constraints on the connectivity matrix because you want these patterns to be fixed point attractors. And these constraints typically, in a network of binary neurons, these constraints take the form of hyperplanes in the space of connectivity matrices. So, for instance, here schematically you have just a network with two synapses, and for a given pattern that you have to learn, the connectivity matrix have to be on that side of this hyperplane. Now, what Gardner did is to compute the typical volume of this subspace of solutions using the replica method. So clearly, as you increase the number of patterns that you have to store, this subspace will shrink progressively until at some point it will shrink to zero. So if you have too many patterns that you need to store, then there are no longer any connectivity matrix that can satisfy all the constraints. And so you can compute this maximal capacity, which you can think about it as an upper bound that's valid for all possible learning rules. So any learning rule will have a capacity that is lower than this upper bound. In particular, for random unbiased patterns, this maximal capacity is equal to 2. So note that it's much larger than the capacity of the Hopfield model. So the Hopfield model is very suboptimal if you compare this with this upper bound. However, interestingly, when you go to the sparse coding limit again, so you can do this calculation in the sparse coding limit, and the scaling as a function of f turns out to be the same one as this as the Sodix-Feigenman model. So basically, this very simple plasticity rule, this very simple Hebbian rule, which is far from optimal when the coding is dense, becomes closer and closer to the upper bound when you go to the sparse coding limit. All right. So this class of models make a number of experimental predictions on two very different timescales. So in fact, these models assume a separation of timescales between the neuronal timescales that are fast timescales. Neuronal time constants are in the range of tens of milliseconds. And these are the timescales of memory retrieval when you're trying to retrieve a particular memory. So in these models, the assumption is that the dynamics is converging towards the local minima of the free energy. And then you have a completely different timescale, which is the timescale of changes in synaptic connectivity, which are assumed to be much slower on the timescale of seconds to even years. And so these are the timescales of learning and forgetting. So on the fast timescales, the predictions of this model is that basically this model, in this model, you have the network converges to an attractor state, which is correlated with one of the stored memories. And the idea is that this is a stable state of the system. So in the absence of other external input, the system can stay there stably over long timescales. And so, and in fact, this can be a mechanism for short-term memory. When you need to maintain information in short-term memory, then these networks will be very good for doing this because the network is basically has settled in a stable state. Another prediction is that in these conditions, the network state should be robust to perturbations because you have these large basins of attractions around the attractor state. On the slower synaptic timescales, the idea is that with repeated presentation of a stimulus, you should modify synaptic connectivity because that's the way by which you store information in these networks. And in turn, this should lead to changes in the dynamics of the network, right? And these changes should be consistent with Hebbian plasticity. And finally, another prediction that these models will make is they will make predictions about the statistics of synaptic connectivity. And if you observe, if you can measure synaptic connectivity between neurons, then the statistics of this connectivity should be consistent with this type of learning. So let me first show you briefly data that is consistent with these two first predictions. And there's been an increasing amount of data that is consistent with attractor dynamics in various behavioral conditions in the brains of various animals, notably monkeys and mice. So in monkeys, there is a long line of experiments that investigated the mechanisms of short-term memory. And for these, people have been using very simple short-term memory tasks, which are also called delayed response tasks. So the idea of this task is that you first showing a particular stimulus to an animal. So here, so this is a setup of a typical experimental setup for these experiments. And one experiment is the delay match to sample experiment, where you show a first image to a monkey sitting in front of a stimulus screen. Then this particular image disappears. There is a delay period during which the monkey needs to maintain in short-term memory the identity of the image that was presented. And then after this delay period, you show, in this particular case, there are several variants, but in this case, you show two images. One is the match, the one that basically matches the sample, and the other one is a distractor. And the monkey has to indicate in some way which one was shown, which one is the sample. And various groups have identified in various brain areas. So in this particular case, these are experiments done in IT cortex, which is a part of the brain which is thought to be the repository of long-term visual memories in particular. What these authors found is that when you present repeatedly specific images to this monkey, so these are the images they were showing to this monkey, so in this particular case, abstract images, then in this delay match to sample task, when you present one particular image to a monkey, so this is an image shown here at this point in time, some neurons are strongly activated by this image, and then they maintain stably a persistent activity, so they maintain stably a high activity level during the whole delay period in response to that particular image. This activity is very selective. It's only present for a small fraction of these images. For example, for this particular neuron, it has a strong activity level, a persistent activity level in response to these two images shown here, but not for other images that are shown in this experiment. So that's consistent with the system that's converging to an attractor state that is different for each of the images that is presented in this experiment. This phenomenon of persistent activity has also been shown more recently in mice. So in mice, of course, you can't do the same experiments as in monkeys. But so this is a much simpler experiment here, where you have just two possible stimuli. And the two stimuli are just auditory stimuli, two different tones, a high tone and a low tone. So the mouse here is head fixed and has two water ports in front of it. And then the mouse learns that if there is a high tone, then the water will appear on the left port. And so it will have to lick left. Otherwise, if it's a low tone, then it will have to lick right. And between this sensory stimulus and the response, there is again this delay period during which the mouse has to maintain the information about the tone in short-term memory. And again, recording neurons in a specific brain area here, where these authors showed that it was restricted to an area called ALM for anterior lateral motor cortex. This is a view from the top of the brain of a mouse. So you have to record in this area. You see that there are neurons that respond, that have a high firing rate during the delay period, but only when, for this particular neuron, the low tone was presented, but not the red tone. So it's consistent with the system having two fixed point attractors, one corresponding to the high tone, one corresponding to the low tone, and the dynamics converges to one or the other, depending on the stimulus that is presented. Now, in mice, one can use, nowadays, one can use new tools using optogenetics. So what is optogenetics? Now, people can make neurons in specific parts of the brain express light-gated ion channels. And so with these light-gated ion channels, you can actually activate neurons with the light. So that's very powerful. And what you can do in particular is you can activate inhibitory neurons that tend to shut down the activity in a specific brain area. And so the prediction here is that when you, so this is a very good way of perturbing a network, and then you can adjust the intensity of light so that half of the time the mouse will give the correct answer, but half of the time it will give an incorrect answer. And basically the prediction of attractor models is that when you perturb the network, it should either come back to the unperturbed trajectory or it should go away and go to some other state. Okay. And so that's here one example neuron where you see for unperturbed correct trials. So this neuron increases its firing rate in response to the blue tone, but not the red one. And here you do a perturbation during this period here. This is this blue interval is the period in which there is the light that's present, that's shutting down the activity of this network. And after the end of the perturbation, the network comes back to the unperturbed trajectory, as you can see here. However, sometimes the mouse makes a mistake, so there is an incorrect trial. And for those trials, in fact, you see that the network is not coming back to the unperturbed trajectory. So consistent with the fact that now the perturbation was strong enough to switch the network to another basin of attraction, and that's why it's giving the incorrect answer. And this is just a couple of single neurons, and this is more like a population measure, which is kind of similar to an overlap, showing essentially the same phenomena. So here, this would be the overlap with the state that the network reaches at the end of the delay period when you're presenting the blue tone. And when you have perturbed correct trials, basically the network comes back to the unperturbed trajectory, but not when you have incorrect trials. Okay, so and there is generally increasing evidence for this phenomenon of attractor dynamics in various experimental conditions and in various behaviors in various animals. So let me now move to dynamics on the slower time scale. So and dynamics at the scale of synaptic changes, which are thought to be much slower than the neuronal timescales. Okay, so there are actually a number of experiments that have shown that if you present repeatedly one particular stimulus to an animal, then the response of neurons to that particular stimulus will gradually change over time. Okay, and which is consistent with the idea that there is some plasticity happening in the underlying network that leads to these changes. So a very well-known phenomenon that's been characterized again in many animals and many experimental conditions is that the average activity level in the brain gradually decreases when a stimulus becomes more and more familiar. Okay, so if you see a novel stimulus, then typically your brain will be more active than if you see something that's highly familiar, and that makes a lot of sense. So this is one of the early papers documenting this effect. You see one stimulus. For the first time, you have a strong average firing rate here, about 30 spikes per second. But these points here are successive presentations of the same stimulus, and you see that this average response is actually decreasing over time. Note that this is like basically something like 3 or 4 hours. So this is something that's happening over quite slowly. This is a whole experimental session that is shown here. Another phenomenon that's been documented more recently is that so you have a decrease of average responses, but interestingly, if you take, so let's say you record the response of a neuron to many different images. So these are recording in visual cortex, in IT cortex. Now you can record the response of these neurons to many novel images, and you can also record the responses of the same neuron to many familiar images. And this shows the distribution of the responses of this neuron to novel and familiar images together. Note that this is a logarithmic scale here for the firing rate. These distributions typically tend to be quite close to log normal distributions. And what you can see is that, as shown in this figure, the average response to familiar stimuli are lower than the average response to novel. But interestingly, what you see also is that the strongest responses, so the strongest responses that a neuron has, are typically for responses for familiar stimuli. So the idea is that the plasticity process that's leading to this. So the strongest responses are responses to familiar stimuli. This is the blue histogram here are histograms. Maybe I did the opposite. So the strongest responses that you see are for familiar responses to familiar images. And so the plasticity process so the idea is that this distribution of responses, the difference between these two distributions can be potentially explained by a synaptic plasticity rule that has modified the network such that the responses to familiar stimuli, the statistics of the responses to familiar stimuli is now different. And this plasticity process should do two things. It should decrease, it would push the bulk of the distribution of responses towards lower rates, but at the same time, it should stretch the tail of the distribution, and so leading to more high rate responses. And so we asked the question of basically what kind of plasticity process would be consistent with these two observations. And so this is work actually done back when I was at the University of Chicago, when we collaborated with these experimental groups that recorded this data. So now we have to reproduce this data quantitatively. We need to move beyond binary neurons, because as you can see, these responses are far from being binary. In fact, they are not even bimodal. The distribution, especially distribution in response to novel stimuli, are well described by a log normal distribution. So we move to a kind of another standard model in computational neuroscience, are so-called the rate models, in which neurons are now described by analog variables that describe their firing rates. And firing rates in neurons typically range from 0 to maybe about 200 spikes per second or so. This is the typical range of firing rates. In this model, firing rates evolve according to this simple ordinary differential equation, where this is the a decay term, that's basically the rates of the neuron will go to 0 in the absence of inputs. And then these are the synaptic inputs to a neuron, which are composed of two terms. These are external inputs, and these are recurrent inputs coming from the same network. And again, this is there is the synaptic connectivity matrix Jij here. And this function phi is a nonlinear function that transforms the synaptic inputs into firing rates. And that's often taken to be a sigmoidal function. Now, what we need to include in this model is how learning happens, right? And so what we did is the following. So when a novel stimulus is presented, what we did was basically draw the external inputs randomly, but we have to do it in a way so that the distribution of firing rates in response to this stimulus should match the experimental data. And so a simple way of doing it actually is to take a Gaussian distribution, IAX to be Gaussian, and then we can choose phi, this nonlinearity, to match the distribution of rates. For instance, if you have a distribution of rates is a log normal, then phi should be an exponential, basically, transforming a Gaussian into a log normal. But of course, they are not exactly log normal. So in fact, we find something that's not exactly an exponential. And in fact, we see signs of saturation for high rates. So that's the way in which we generate the rates of the responses of the neurons in the network. Now we need to choose a way to change connectivity, synaptic connectivity in response to the stimulus. And we choose an unsupervised plasticity rule. So what does unsupervised means? It means that the only information that synapses have to change their are the firing rates. So the only information that synapses have to change their states are the firing rates of the presynaptic neuron Rj and the postsynaptic neurons Ri. So this is basically a kind of basic locality constraint. Synapses only know what the two neurons they are connecting are doing, but not what other neurons are doing. For simplicity, we assume that this function is actually a separable function of the presynaptic firing rate, so through this function g, and times the function of the postsynaptic firing rate, this function f. All right, so now if we choose this function f and g, we can now change the connectivity matrix, and now we can again present the same stimulus. You can use the same IIX, and then compute the distribution of rates for that once the connectivity matrix has been changed. And in the model, that gives us the distribution of rates for familiar stimuli. We assume that any stimulus that has changed the connectivity matrix is now familiar. And of course, now the goal is to find delta J such that the distribution of rates for familiar stimuli will match the empirical distribution, the one that we see in the data. So let's make some kind of simple back on the envelope calculations to just have an intuition of what's happening here. So if we're changing this connectivity matrix, we'll have changes in the total synaptic inputs that neuron receives that will be proportional to this this equation, right? You have a change due to Jij changes, so you have a basically sum of the J, delta Jij, Rj. Of course, the rates also change, so you will have some other terms there, but we don't need to care about these terms now. And we use the fact that it's a separable function, and we see that basically the function of the postsynaptic firing rate factorizes, and we see that this delta Hi is just f of Ri times a constant, essentially, which is basically the average of G of R times R over the whole network. So basically, how the synaptic inputs to a neuron will change after you learn a stimulus depends actually on this function f. And so it depends on the visual response to that novel stimulus through this function f. In particular, the sign of f will determine whether the response of that neuron will increase or decrease as you're presenting this stimulus repeatedly. And in particular, let's take a simple example just to see what happens. So we can take a popular plasticity rule, is a covariance learning rule. In fact, the covariance rule is the one that's used basically in the Hopfield model or the Sodix-Feigelman model. You take the product of the rate of the post minus the average rate times the rate of the pre minus the average rate. And basically, it's very easy to see that the changes in synaptic inputs will be basically proportional to the deviation of the rate of the postsynaptic neuron minus the mean times the variance of the rates. And let's take another simplifying assumptions with a linear transfer function. That means that the mean change in rate should be 0. Okay, but individual neurons, of course, should change their rates. In particular, if the visual response initially is higher than average, then the rate should further increase with learning. If it's lower than average, it should decrease with learning. So essentially what you will be doing is broadening the distribution of rates. You start with a distribution like this, then with familiarity, it will become broader and broader. So we see that this learning rule doesn't quite capture the data very well. In particular, it doesn't reproduce the decrease in the mean response that we see in the data, though it does capture the fact that you get stronger and stronger. So it does capture the evolution of the tail on the right tail of the distribution, right? All right. So basically, we can, of course, using this insight, we can actually figure out what is the function f that matches the data. So it's kind of a little bit complicated procedure involving different quantile functions that I don't have time to describe in detail. But the main result that we find is that so neuron by neuron, we can infer basically this function f that by construction will map a particular novel distribution to a familiar distribution. So for this particular neuron, for instance, this is the function f that we find. As you can see, this is very well described by a sigmoidal function, which is negative for rates that's lower than about 30 spikes per second here, and becomes positive for high rates. And the intuition here is that this negative part leads to the decrease of rate of the fact that the bulk of the distribution is pushed towards lower rates. So that's this negative part. And then this positive part at high rates is responsible for the stretching of the tail on the other side. Okay. And interestingly, we see that compared to the covariance rule, there are two major differences. So first, it's highly nonlinear. And the second major difference is that the transition from negative to positive changes is actually occurs at a threshold. Here you have a threshold for between depression and potentiation that's actually a few standard deviations above the mean rate. So that means that for the vast majority of stimuli that are presented, the rate will actually decrease, while for a small fraction, they will increase. It's that typically for most of the neurons in this data set, it's only a few % of stimuli that are above this threshold. In fact, for many neurons, for some neurons in the data set, we don't see any in any positive region, but we speculate that maybe because there were not enough stimuli that were used in this experiment, that if they had used a larger stimulus set, then maybe we would have found this region. So the main message here is that what this rule is doing is actually sparsening the representation of these external stimuli with learning, because you're pushing the vast majority of neurons have their rates pushed down towards zero, and only a small fraction of neurons see their rates increase. And actually, that makes a lot of sense if we come back to the original models that I described, which were these early studies found that actually storage capacity typically increases with a sparser coding. All right. So the next step was actually to study the dynamics of networks where these synaptic plasticity rules are constrained by data in the way that I showed you. So basically, we have basically the same model that I already described, and as I've already mentioned, we can also constrain the transfer function using data. And basically, this is an example of transfer function. So this is basically this procedure where we're taking I to be a Gaussian, the external input to be a Gaussian, and then we can compute phi by matching such that we will match the distribution of rates for novel stimuli. We get something like this, and we can fit it with a sigmoid. And likewise, we have the connectivity matrix, which is also constrained by data. G actually cannot be fully constrained by data in this, but we just assume that it's also a sigmoidal function as F. and basically, we can build the model in a similar way as in the Hopfield model. We can take a linear superposition of patterns, except that now this model is, of course, much better connected to this experimental data. In fact, by construction, it reproduces this distribution of firing rates that are observed in the data. And so we started to study this network. And the first thing is one question is, can it actually produce attractor dynamics? And in fact, the answer is yes. So this is a simulation of a network where we can start from a state that's uncorrelated with all the patterns that are stored in the connectivity matrix. In fact, that's one difference with the Hopfield model is that, and actually it's a feature also of this Zodix-Feigenman model, is that this class of models, essentially when you break the symmetry between plus and minus one states, they have typically a kind of ground states where all neurons are at low activity states. So that's this kind of, we're starting from this baseline state. And then we present one of the patterns that are stored in the connectivity matrix, and you can see that then after the stimulus is removed, the network converges to a state that's different from the baseline state. And of course, it's this, as in the Hopfield model, this state will be strongly correlated with one of the patterns that's stored, basically the pattern that was presented. And so we have this picture that's similar to what we have in the Hopfield model. And interestingly, something to emphasize is that the plasticity rule that we inferred it using data from IT cortex of primates, and that's the same area where people have documented this phenomenon of personal activity. So this is very consistent with these observations. Okay, and in fact, in this model, we can also use kind of standard tools. So you can use mean field theory to compute the storage capacity in this model. In particular, we used, for simplicity, we used a sparsely connected network such that to facilitate to make these calculations easier. In particular, there is, in fact, the connectivity matrix in this model is actually not symmetric, so we could not really use in a fully connected network. We would not be able to use the replica method, but we can use in a sufficiently sparsely connected network. We can actually use methods similar to the paper of Derrida et al. And I don't want to spend too much details on this, too much time on these calculations, but essentially what we can derive are equations for the order parameters of this network. So we have one order parameter it describes the overlap with the patterns that you're trying to retrieve. So when you're presenting one of the patterns, so let's say the first pattern that was stored in the connectivity matrix. So this parameter M will basically describe how strongly correlated the network state is with that particular pattern. Note that in this model, the order parameter is actually a nonlinear correlation because of these nonlinear functions that imprint the patterns in the connectivity matrix. And then you have this other order parameter, which basically describes the noise. So when you're retrieving one pattern, the thing is that you have the other patterns that are stored in the connectivity matrix that are kind of interfering potentially with the retrieval of that pattern, of that memory that you want to retrieve. And so that's captured by this order parameter sigma square. It's a kind of noise term. This is a kind of signal term, and it's kind of noise term. And we can derive these equations in these limits, large N limit, and also sparse connectivity limit. And basically, the idea is to find solutions of these equations such that this overlap is a non-zero. So these are retrieval states. So you, of course, to be able to retrieve the memory, you should have a non-zero overlap with that particular memory. Otherwise, you can't really retrieve it. And so we can find these solutions numerically. And we can also compute the storage capacity, which is basically corresponds to the largest value of alpha. So alpha is again the ratio between number of stored patterns and the average in degree of this network. So C times N, C is the connection probability. And so the storage capacity will be the largest value of alpha for which you find non-zero solutions of this equation with non-zero N. So for parameters that are basically inferred from data, what we find is that we find a storage capacity which is about 0.6. Okay. And so what we ask then is we ask the question, so is it good? Is it bad? And in particular, we ask the question, what would be the best, what would be the best functions F and G in terms of maximizing storage capacity? What are the functions F and G that maximize storage capacity? And so we search in the space of these functions F and G. So we constrain this search a little bit. We really search in the space of sigmoidal functions F and G. And we found that actually the functions that optimize capacity are actually when F and G are just step functions with high thresholds. And it turns out that the model then becomes basically very similar to the Sodix-Feigenbaum model, because essentially for step functions, what you're doing is, so you have initially a Gaussian input patterns that are transformed by the neuronal transfer functions become log normal, but then you're passing through step functions, so you're binarizing these inputs prior to storing them in the connectivity matrix. And in fact, interestingly, so it turns out that the functions that we infer from data are very close to step functions. As you can see here, this is a sigmoid with a very high that's actually very close to being a step function, and it also has a very high threshold. So that's why the capacity that we are finding is actually pretty close to the optimal capacity that in the space of this function F and G. So that's kind of an interesting and I have to say that quite unexpected result. Just want to mention briefly that we can explore parameter space in this class of models, particularly increased. We can, instead of using parameters inferred from data, we can basically change the coupling strength in this model by basically adding this constant A and increasing this constant A. And we find interestingly that when we increase the coupling strength sufficiently, there is actually a transition to chaos. But interestingly, what happens is that you have now the retrieval states are no longer fixed points, but they become chaotic. Okay, so now the space of states of the network look like this. So you have now these different regions in state space where the dynamics is chaotic, but the dynamics remains confined to a region which is still close to each of the stored memories. Okay, so this shows here a simulation of this network. You have these wild fluctuations of the firing rates of all the neurons. But note that basically, after you presenting one pattern, you see that you can already see by eye that this chaotic state here seems different from the background chaotic state that you see here. And in fact, this is this overlap that measures the quality of retrieval that you can measure in this simulation, and you see that this overlap is actually there is actually a non-zero overlap with the memory that is being retrieved here. So that's an interesting an interesting behavior that you see in this model. In fact, it's been argued. So there has been a number of observations, in particular in monkeys, that many times in these memory tasks, people see activity during delay period that is strongly irregular. And it's been argued that this is not, some people have argued that this is not consistent with a fixed point attractor scenario because you see this very strong irregularity. And we argue that maybe this is a signature that in fact we have chaotic memory states and not fixed points. All right. So I just wanted to, I know that I'm pretty close to, I only have 5 minutes left. I just wanted to show you a different, briefly, the last part of my talk, which is about trying to derive other types of predictions about the statistics of synaptic connectivity in these networks. And for this, we'll kind of go back to going back to the introduction. I showed you that there was this different approach that was proposed by Elizabeth Gardner at the end of the 80s, and the idea there was not to study a particular plasticity rule, but to study the space of all possible connectivity matrices that are consistent with learning a set of fixed point attractors. So what we've done in several steps is to use the same method, but now to derive various statistics of a connectivity matrix in this volume, in this Gardner volume. And for this, we can again use the toolbox of statistical physics and either replica or cavity method. In some cases, cavity method is more convenient. And in particular, what we are interested in just very simple quantities, like what's the distribution of synaptic weights in this Gardner volume, or what are joint distribution of weights in pairs of neurons. If we measure two neurons, you measure synaptic connectivity in both directions. What does the joint distribution look like? And then for this, the cavity method is more is simpler, and we can use so-called two-side cavity, where you're just adding two weights to a network that has all. And so using the basic ideas of the cavity method. Now there is some important constraint that we need to take into account. If we want to compare this statistic with data, we have to implement a very important constraint that's present in real synapses. And this constraint is that in real synapses are sign constraints, right? Synapses can be either excitatory, or in other words, positive or zero, or they can be inhibitory, but not both. And they cannot change with learning. And so here in this picture, that means that we have these additional constraints that we need to take into account, which here are basically that we are in this simple toy cartoon of this network with two synapses. Basically, we are confined to this upper right quadrant, basically. So you have additional hyperplane here, hyperplanes that limit the space of weights. And so there are just a couple of interesting results that we find using this approach. So we find that the distribution of weights that at maximal capacity, so we are really interested at what happens when we are close to the point where this volume shrinks, and at that point everything simplifies. In fact, all these distributions become basically truncated Gaussians, where they are much more complicated if we are far from maximal capacity. And at maximal capacity, these distributions simplify. They are just sums of delta functions and the truncated Gaussians. Intuitively, you can think about these distributions as being a Gaussian, and then you remove the negative part and you put it at 0. And interestingly, so that essentially what that means is that when you reach maximal capacity, the network will be no longer fully connected, but there are a large fraction of synapses that are actually no longer there. Okay. Or they have 0 weight. And so effectively you have a connection probability that's due to learning there. So this is really an outcome of learning. And an interesting result is that connection probability is basically less than 50% for any parameters of this model. And it decreases with the robustness of learning. So you can do these calculations imposing a particular degree of robustness to learning. So there is a robustness parameter that will tell you how big are the basins of attraction around the patterns, and we can see that the connection probability will decrease monotonically with this robustness parameter. The other, and I don't have time to tell you about this geometric interpretation of this result, but the other interesting result that we find is that when we compute the joint distributions of pairs of weights if we have just 2 neurons, what is the joint distribution of weights in both directions? It turns out that the outcome of the calculation is the correlated to the Gaussian, a truncated correlated to the Gaussian. So this is an illustration here where we see this Gaussian, and basically you have delta functions on the axis and the product of 2 delta functions at 0, 0 here. And the correlation is due to the fact, of course, that basically these two synapses, they are quite strongly correlated by the fact that they see the same patterns of the pre and postsynaptic neurons. But interestingly, one might have thought that if you want to store fixed point attractors, you might want to have a completely symmetric connectivity matrix, in which case basically the distribution would be basically concentrated on the diagonal. But it turns out that it's something intermediate between a fully symmetric and a fully asymmetric matrix. And in fact, so if we plot the results on a particular way to plot the results would be on this plane where the x-axis is connection probability and the y-axis is the reciprocal connection probability. So the probability that neurons are connected in both directions. Here, the diagonal would be basically a symmetric network. Here, this is the y equal x squared for asymmetric, random asymmetric network where the two are uncorrelated. And basically, these are the curves that are obtained by varying this robustness parameter for different coding levels, right? So this is 0.5, 0.1, 0.01. You can show that very, very slowly it will approach this symmetric line when the coding level becomes really small, but extremely slowly. And this is actually data. So this is the largest data set that we could find by Song et al. From recordings from rats, and the data is actually here. In fact, that's something that has been noted by many experiments, is that connectivity in cortex there is this strong overrepresentation of bidirectionally connected pairs of neurons compared to a random Erdős-Rényi network. So that's a very common observation that people have seen in most cortical areas. Interestingly, not in all. So there is the exception of the mouse barrel cortex, which is basically the part of cortex that receives information about the whiskers, which is the whiskers, which is a very important sensory input for rodents. All right, I think I should conclude. I just told you part of the story. So this is another another way to try to make a contact between these theoretical models and data. And in fact, there is increasing data that's becoming available, both recordings of neuronal activity, but also recordings of synaptic connectivity. So there's a whole new field called connectomics, where people reconstruct a whole neural circuits and the whole connectivity matrix of neural circuits. And now it's recently the whole wiring diagram of Drosophila have become available, and of various species of the mouse brain. And so now it's becoming possible to test some predictions of these models. And so there are many open questions that we're interested in. I just want to mention that we're studying here with Marco and Enzo. We are studying the learning process. So this is a continuation of the first part of my talk. And we have some interesting results about what happens there. There is, from connectomics data, there are projects on how you can infer, if I give you a synaptic connectivity of a network, so these are now becoming available, can you tell what are the memories that are stored in this network, basically? And so that's kind of a very interesting inference problem that we have some preliminary results with Sebastian Gold, Florent Kerzakala, and Lenkas Deborova. And in fact, I actually gave a talk in this room 2 years ago about this particular problem. But there are, in fact, there is new data that have recently appeared that I think would be very interesting to analyze. And finally, these are the people who've been working on the first part of the talk, and so Subin and Ulises in particular, and the experimentalists. Yali, who's collaborated, is actually the son of Daniel Mead. And these are the experimentalists with whom we've collaborated on this project that provided the data that I showed you. Thanks for your attention. I'm happy to answer any questions.
Thank you very much for this brilliant talk. So, are there any questions? Also, if people from connected can also ask questions, so you can be listened everywhere. Are there questions or comments?
The first question is, in your model, you always consider random patterns or any correlation between patterns?
Right. So personally, I've always considered random patterns because, of course, that simplifies everything. So there is a long literature on what happens when you have correlated patterns. So that, and so starting from Cantor and Sopolinsky back. So then of course, you can no longer, if you use the very simple learning rules that I showed you, it's basically a disaster. And the capacity basically goes to drop to zero as soon as you have correlations. So you have to use other rules. So of course, you can use something called a pseudo inverse, and then you're fine. But the question is, how can you implement it in a neurobiologically plausible way? And so there are all these ideas, maybe unlearning, all these things, which, these are just, at this point, these are just theoretical proposals on how to store correlated patterns. In fact, I think so the one question that is still not completely clear is in areas which are in which memories are stored, are there really significant correlations in the stored patterns? Or one could imagine that basically sensory systems that lead to these areas are actually tried as best as they could to decorrelate these inputs so that they could be more easily stored in memory. So that's one scenario. The other scenario is that you still have significant correlations, and then you need to find a way to deal with these correlations. And I think there are proposals for how to do that, but I think it's fair to say that it's not clear whether the brain really implements these proposals or not. So, yeah.
I have also a second question. Regarding the topological properties of the network, you said that you analyze the red point in your plot was when you made the largest data set that you find. So my question, the first question is how large it was. So how many neurons people are able to map exactly the connection. And the second question is, if one wants to go beyond the pairwise connection, if people also compute the next level, which is, say, the triangle.
So okay. So these the experiments that are the data that are showed are basically come from these kind of experiments. You take a slice of the brain, and then you record n neurons, and so it depends on the skill of the experimentalist. In this case, 4 neurons simultaneously using these pipettes. So you see these 4 nice neurons here, and then you can basically look at you can compute the connectivity matrix between all these neurons. You stimulate each neuron at a time, and then you record the others. And for example, here in this little 4 subnetwork, you have just 2 connections that exist out of the 12 possible ones, and they happen to be between the same pair of neurons. Okay. In this experiment, to give you an idea, so Jesper Sjöström, he had recordings of something like 20,000 pairs of neurons. So it was basically during recordings during a very long time. So out of these 20,000, you have only 2,000 connected pairs because the connection probability is about 10%. Okay, but basically at this level, this is actually pretty. The estimate is quite precise because you have so many pairs. Now they also looked at distribution of triplets, but now triplets, of course, you have like 16 possible triplets. They found an overrepresentation of strongly connected triplets compared to a network that would have the same pair statistics. So you can take a random network and observe pair statistics, and you still have overrepresentation of strongly connected triplets. People have also looked at, so the record number is 12 simultaneously recorded neurons by Henry Markram. You might have heard about him from Lausanne. And then, of course, so the number of motifs explode. So it just looks at how many. So they just look at the probability of observing a certain number of connections in these networks, right? But it's consistent with this kind of clustered DST idea that you have clustering these networks, so you have higher representations of strongly connected subgroups of neurons compared to random. Now the thing is, so now that you have this other method, which is this connectomic approach, where you reconstruct the whole cortical area, and now there is a data set that's available where they basically, in principle, you should have the connectivity matrix of the whole cubic millimeter of mouse cortex. And so that's kind of my to-do list, to kind of look at this data, and then basically that's an enormous amount of data that you can analyze.
Okay, we have 5 more questions. Yes, I know it doesn't. Okay, so the first one is from Pier Stanislao Paolucci, that has a couple of questions. Can you ask?
Yes. Do you hear me?
Yes.
Okay. So thank you very much for the very interesting talk, and for the full coverage of the historic results and novel ideas. So now my question is, are you considering in any manner the effects of sleep on the storage of the memory, in particular the generalized homeostatic depressive processes that are, however, associated with the creation of association among similar memories? And this is a first question. And the second one, the fact that indeed neurons project either excitatory or inhibitory synapses, but not both. So if you have consideration about how to include this in this kind of models.
Yeah, great questions. So the first question about sleep. So in this work, when we inferred plasticity rules from data, so we just compared 2 types of images, right? So the novel ones, so never shown before, and the familiar ones were shown like many times over multiple days or even months. So it's hard to, given this, it's impossible to disentangle what are the effects of presentations during the day versus what might happen during the night, where there have been various proposals. Maybe you have reactivations of memories during the night. As you mentioned, there are various homeostatic processes occurring. And so we can't really address these questions with this data. But now we have this ongoing collaboration with this group in Chicago, where they actually, now they are following, so they have the same set of images which they were presenting day after day. And they are looking at what happens during one day, and then you can look at what happens like the next day when you're starting the experiment. And so then maybe there is a hope to disentangle different effects of what happens during the day and possibly during the night. But this is still, we still haven't fully analyzed what's happening. Second question about excitatory and inhibitory. So in this part of the talk, so I haven't mentioned this because I didn't have time, but the idea is that all the neurons that I'm showing here are what they call putative excitatory neurons. And so this really goes into the details of the experiments, but basically these are extracellular recordings. They record the spike waveform. So this is the electrical signature of action potentials. And you can distinguish, or some people claim they can distinguish between excitatory and inhibitory based on the waveform of the spikes. In particular, the excitatory neurons are supposed to have a broader waveform, and some inhibitory neurons, the fast spiking ones, have a narrower waveform. And interestingly, this kind of this type of behavior, you see it only in putative excitatory neurons, but not inhibitory. So inhibitory neurons, when you apply the same procedure, they will be consistent with basically having a completely flat curve.
Okay.
So that's one. So one possibility is that this type of plasticity in this particular case, or at least the Hebbian component of plasticity, is only implemented through excitatory connections.
Okay.
But of course, inhibitory, you mentioned homeostatic processes. Some people have argued that you could have inhibitory plasticity that actually implemented some kind of homeostatic process to stabilize the network, in particular, that could help to counterbalance instabilities that can arise when you have Hebbian learning. So that's certainly possible. We can't really, the analysis that we did does not really provide any answers.
Okay. Okay. So thank you very much for the answer. In our lab here in Roma, we have spiking stimulation that makes learning using an STDP Hebbian mechanism, and then we have evolution with sleep and REM models. So I think that it will be really interesting if we could contact you and we could compare what happens in a rate-based model and in our spiking models. So.
Okay, well, I'll be very happy to talk further. Sure.
Okay. Thank you very much.
Okay, we have another question from remote by Silvia Scarpetta.
Thank you. Thank you for the nice talk. I have a question about the learning rule that you infer from the rate data. Have this learning rule a relationship with the spike time dependent plasticity that is observed at the neuron level?
Mm-hmm. Yeah, another very good question. So, yeah, I think there could be like a whole other talk about different types of plasticity rules, spike timing versus firing rate based rules. So, I mean, the short answer is that is no. So there is no spike timing information at all here because we are considering a rate model. And so interestingly, when you look at experimental data, so people have tried to induce plasticity in brain slices, and we mentioned spike timing dependent plasticity. So the plasticity depends on the relative spike timing between neurons, but it also depends on the firing rate, something that's perhaps less well known to some people. So typically, the idea is that synapses are sensitive both to the timing of spikes, but also the rates. And this plasticity rule, actually, I'm actually on the right slide here. This plasticity rule is actually quite consistent with recordings with plasticity experiments in vitro that change the rates of pre and postsynaptic neurons, and they show that at low rates, typically you see depression, while at high rates you see potentiation. Now, in addition, so you're right that synapses are also sensitive to timing. So if you have pre-firing before post systematically, then you tend to get more potentiation and more depression in the opposite order, but that's not incorporated here. We have a separate line of work in which we take into account this spike timing dependency. So essentially what that does is that leads to a temporally asymmetric plasticity rule, where now, so in particular, if you have temporally varying inputs, external inputs, that can lead to a strongly asymmetric connectivity because of this spike timing effect. And we've worked on how you can generate, in particular, sequential activity networks or different types of activity using this type of plasticity rule. But this is ongoing work. But yeah, thanks for asking the question.
Okay, thank you. So you measure at the same time here, FDRI and GDRJ at the same time, not with you don't consider correlation between some milliseconds.
Right. So we only here we only analyze the average firing rates of neurons during presentation of the stimulus. So we didn't look at the spike timings, and this would be this is something that we should do at some point. We should look at whether there is any, especially so something that we do find is that when we look at the dynamics of the response on fast time scales, so when you look at the firing rate as a function of time, there is actually no difference in the firing rates in the early phase of the stimulus presentation. Okay, and so it looks like there are no significant differences in terms of at least in the early phase of the response. There does not seem to be systematic differences in the response to novel and familiar. What one might have expected that with spike timing dependent plasticity, the network will respond earlier to familiar stimuli. So there have been people have argued that could happen with STDP rules, but we don't find any indications for this effect.
Thank you very much.
Okay, we have another two questions. One is from the audience.
As usual. And now the question is, from what you were telling us, in the end, plasticity is always at work. It never stops. So can you comment about that? So the synaptic matrix stabilizes at some point, or every time you are learning something, actually you are destroying rest. Shouldn't you expect an F function that is stopping plasticity, for instance, at low rates or something, some mechanism like that?
Yeah. Yes. So right. So in particular here, so that's a very good observation that you're making, that in this plasticity rule, we have also, if the rates are zero, you will have some changes in synaptic efficacy. That's clearly not realistic if one thinks about this plasticity rule exclusively in terms of Hebbian plasticity, where in principle, if two neurons are inactive, there shouldn't be any changes. However, what we can do is we can decompose this delta J in two terms: a Hebbian term, which would go to zero at when both rates are equal to zero, plus a term that is non-Hebbian, which could be a stabilizing term that ensures that in average the synaptic changes are actually equal to zero. So in that interpretation, we basically, what we're measuring here is a combination of multiple plasticity processes. There is a Hebbian process, and you're right, it should go to zero at zero rate when both rates are zero. And then there is another term which is more homeostatic in nature. So that's how I think we should think about this. Yeah.
Okay, and the last question is from Roberto Livi. Roberto, are you there? Roberto, we can't hear you. We cannot hear you. Okay, I can read your question. Okay. Is does the transition to chaotic attractors modify the recovery timescale of patterns? And in case, how?
The recovery timescale. Good question. In both cases, you have a pretty fast. So the network actually converges pretty fast to these attractors, whether they are fixed points or chaotic. But we haven't really compared systematically the times of convergence in these two cases. Yeah, but that's something we should investigate.
Okay, if there are no other questions, we thank our speaker again. And goodbye, everybody. Thank you for being here.